Hi, my name is Emily Matthew, and today I'm going to be talking about a novel class of drugs called CDK PROTACs and its potential to more effectively treat breast cancer. So, just a little bit of background on the disease which I focus on in my research. I'm sure many of you are familiar with breast cancer, but breast cancer is a very aggressive and very prevalent disease throughout the world, with 2.3 million women currently diagnosed with it and over 670,000 women who have already died from it. Globally, about 1 in 4 women is diagnosed with breast cancer, and nearly 1 in 8 women die from it. And although there are many treatments for breast cancer on the market, many of them become less effective over time due to mass drug resistance. Now, this means that within 3 to 5 years of a patient being exposed to a drug, 30 to 50% of these patients will have built up resistance to that medication, and this renders the medication essentially useless to them. Now, of the current hormonal treatments for breast cancer in the market today, they can be essentially divided into two main groups: inhibition-based drugs and degradation-based drugs. Inhibition-based drugs, like aromatase inhibitors and selective estrogen receptor modifiers, or SERMs, approach the treatment of breast cancer by using inhibition-based techniques. But as I mentioned before, these types of drugs run into the issue of acquired drug resistance, which renders them useless. So researchers have shifted to a new type of drug class called SERDs. SERDs, or selective estrogen receptor degraders, focus on a similar technique as SERMs, except instead of inhibiting overexpressed estrogen receptors, they degrade them. And although this is a much more effective strategy compared to SERMs, SERDs also come with their own set of drawbacks, including the fact that SERDs are not orally available and must be taken as an injection. And also, they have a very large chemical structure, which means that they have a very poor ability to be chemically diversified. And these factors played into the fact that the FDA has only approved 2 types of SERD treatments, and this has limited accessibility to the drug. And all of these factors resulted in the development of a new drug class called proteolysis targeting chimeras, or PROTACs. PROTACs approach the treatment of breast cancer in a similar way to SERDs by degrading overexpressed estrogen receptors. However, PROTACs are able to do this more effectively than SERDs because PROTACs not only degrade the estrogen receptors from one region, but from various regions. And because of this versatility, PROTACs are more effective in treating breast cancer than previous SERD treatments have been. But my research was not only rooted in the field of PROTACs, but also the developing subfield of PROTACs, CDK PROTACs. CDKs, or cyclin-dependent kinases, specifically CDKs 4 and 6, are important enzymes in the cell division process that can sometimes contribute to the growth of cancers. In normal cells, CDK4/6 molecules regulate parts of the cell cycle. However, in cases of cancers, the protein can become overexpressed, which can cause uncontrollable growth and division of cancerous cells. And this means that the stronger the interaction between a drug molecule and the CDK4/6 molecule is, the less amount of drug resistance the patient would result in, and the lower the risk of that patient to have a cancer relapse. And this means that the general field of PROTACs focuses on degrading overexpressed estrogen receptors, while the field of CDK PROTACs specifically focuses on degrading CDK4/6 molecules. Now, in order to test this theory that CDK PROTACs would be more effective in treating breast cancer, I used the Schrödinger Maestro software to not only visualize the structural compounds of the various structures I looked at, but also compute the docking scores of these compounds. And a docking score is essentially an indication of the binding affinity of that molecule to the CDK molecules around it. And this means that the lower the docking score, the stronger attraction and strength of the bond that that molecule would have to the CDK4/6 molecules around it, and the more likely that molecule would be to have degraded the CDK4/6 molecule. So the takeaway is the lower the docking score, the more effective that compound is in treating breast cancer. So just to summarize, the problem that I'm trying to address in my research is the limited amount of treatments to treat breast cancer that are not short-term treatments that have acquired drug resistance and are also not treatments that have a very small, poor oral availability. So the objective of my experiment was to look into the structurally novel class of small molecules of CDK PROTACs to determine which type of structure would be the most effective in inducing CDK4/6 degradation, as well as blocking cancer cell proliferation. And my hypothesis was that as a result of docking scores and binding affinity being inversely related, the more interactions and the stronger the interactions of a molecule with the CDK4/6 molecules was, the lower the docking score would be, and the more effective that molecule would be in treating breast cancer. So the methodology, as I mentioned before, involved using the Schrödinger Maestro software to compute the docking scores. And I computed the docking scores of 7 identical structures that were varied slightly. And just to give you a little look at what the structures of the CDK PROTAC molecules look like, this is the essential backbone of every PROTAC molecule. It has 3 parts. The left-hand side is the protein of interest part, which is a ligand that binds to the protein of interest that's going to be degraded, which in this case was the CDK4/6 molecule. And this means that the left-hand side, including the linker that connects this ligand to the other ligand, was constant in all of the 7 structures because we wanted to keep the same type of CDK4/6 molecules to be attracted to this type of PROTAC. However, the right-hand side of the molecule was adjusted slightly to determine which kind of structures would be the most effective in treating breast cancer. So as you can see from the results table, the chemical structures on the left-hand side, as I mentioned before, are identical in each of the 7 structures. However, the right-hand side of the molecules was altered slightly, and this is what results in the varying of the docking scores seen on the right-hand side of the table. So structure 5 had the lowest docking score of negative 11.484, which meant that structure 5 was the most effective in degrading CDK4/6 molecules, closely followed by structures 1, 2, and 7 based on their docking scores. And in conclusion, structure 5 had the lowest docking score, which meant that it had the highest binding affinity to the CDK4/6 molecules that it was trying to degrade. And this means that if there were to be a CDK PROTAC-based drug, this type of structure would be the most effective in not only inducing CDK4/6 degradation, but also blocking cancer cell proliferation. And once again, I would like to emphasize the benefits that CDK PROTACs can bring compared to the previous treatments that I've mentioned. First of all, it has a lower risk of toxicity and a lower risk of overdosing because of the smaller dosages that CDK PROTACs have, because they are a more effective treatment compared to other treatments that would result in higher doses because they are less effective of a treatment. And also because they use degradation instead of inhibition, it is a more long-term treatment compared to a short-term treatment, as in the inhibitory technique drugs that I mentioned earlier. And knowing all of this, and as I researched this and as I completed all of the experiments involved in my research, I was fascinated by the field of PROTACs and everything that encompassed the general field of PROTACs as well as the subfield of CDK PROTACs. And I realized there are so many things that have not been explored that I would love to explore, including but not limited to the fact that PROTACs have as of now been used primarily in breast cancers and other types of cancers, but can be expanded to many other diseases, and that's such an interesting topic to explore. And also the fact that there is an up-and-coming idea of prefused PROTACs, which I'm not going to get into right now, but is very, very interesting and could potentially increase the rate of effectiveness of PROTAC drugs by nearly 2 times, which is incredible. And these are some of the references that I used throughout my research. And once again, my name is Emily Matthew. Thank you so much for listening to my presentation, and I hope you have a great day.